ATP generation from fatty acid oxidation. The net yield of ATP per 1 oxidized palmitic acid, the number of cycles is 7 according to the equation n over 2 minus 1. We can represent the beta oxidation reaction as this equation. Palmitoyl CoA plus 7 CoA plus 7 FAD plus 7 NAD plus 7 water molecule lead to formation of 8 acetyl CoA plus 7 FADH2 and 7 NADH and 7H. So we can calculate the energy or ATP molecule generated from beta oxidation of palmitic acid by this simple mathematics calculation. 8 acetyl-CoA. Each acetyl-CoA produces 12 ATP by Krebs cycle. So we multiply 12 by 8 equal to 96. Also, each molecule of FADH2 will produce 2 molecules of ATP by electron transport chain, so we multiply 7 by 2, 14 ATP. Also, each molecule of NADH produces 3 ATP by electron transport chain, so we multiply 7 by 3, 21. So the sum of this ATP molecule equal to 131. But we use 2 ATP for activation palmitic acid in order to convert it إلى palmitoyl CoA. So the 131 minus 2 lead to net ATP molecule equal to 129 molecule. Odd-numbered fatty acids are rare. It's only present in some type of plants. So the final round of beta oxidation of this fatty acid yields propanyl-CoA instead of acetyl-CoA. So this propanyl-CoA is converted to succinyl-CoA, which one of the intermediates in a Krebs cycle. Also, propanoate is also produced by oxidation of isoleucine, valine, and methionine. And also, it is the minor precursor of gluconeogenesis. Factors regulating mobilization of free fatty acid from adipose tissue. Liver has a large capacity to take as much as 30 to 50% of free fatty acid from plasma, both in feed and fasted state, but the release of free fatty acid from adipose tissue is more in starvation. Second, inhibitory effect of malonyl-CoA on carnitine acid transferase 1. In feeding state, insulin activates acetyl-CoA carboxylase to form malonyl-CoA. So malonyl-CoA inhibits carnitine acid transferase 1, resulting in decreased beta oxidation. While in starvation state, increase in free fatty acid inhibition of acetyl-CoA carboxylase enzyme activity and then decrease in malonyl-CoA concentration, leading to an increased fatty acid oxidation. So it's regulated by the insulin to glucagon ratio. Now we start to explain the ketone bodies. Are alternative fuel for cell. Liver mitochondria have the capacity to convert acetyl-CoA into ketone bodies. Ketone bodies are acetoacetate, beta-hydroxybutyrate, and acetone. Acetoacetate and beta-hydroxybutyrate are transported in the blood to the peripheral tissue. They can be reconverted to acetyl-CoA, which can be oxidized by Krebs cycle. In fasting or diabetes, The gluconeogenesis is activated and oxaloacetate is consumed in this pathway. So the fatty acids are oxidized, producing excess of acetyl-CoA, which is converted to ketone body, which are the hydroxybutyrate, acetoacetate, and acetone. Ketone bodies are synthesized in liver mitochondria and exported to different organs. Ketone bodies are fuel molecules for brain and other cells during starvation. Early stages of starvation, when last remnants of fat are oxidized, heart will consume ketone body, reserving glucose for the brain. This slide shows the chemical structure of the ketone bodies, which is beta hydroxybutyrate, acetoacetate, and acetone.